Assalamualaikum and hi everyone. My name is Nur Amirah binti Muhammad Suwardi, and today I will be presenting to you guys steps of recombinant DNA. So before we move on to the steps of recombinant DNA, allow me to explain to you guys about what is recombinant DNA technology. Recombinant DNA technology is joining together DNA molecules of two different sources to produce new genetic combinations, and is used in various fields in science such as medicine, agriculture, and industry.
The recombinant DNA are divided up to 5 steps: isolation of gene, cleave or cut, insertion, transformation and amplification, and lastly, screening. The first step, isolation of gene. So what we do in isolation of gene is, we will isolate the DNA vector or DNA plasmid from the bacterial cells and the DNA containing gene of interest from source. Usually, the source of the plasmid is typically E. coli. After the isolation, we'll move to the next step, which is cut the DNA. We will cut the DNA donor and plasmid DNA with the same and compatible restriction enzyme as to produce complementary sticky end or blunt end. The restriction enzyme makes a single cut within the exact gene on vector DNA or plasmid. The restriction enzyme that we will be using is EcoR1. EcoR1 is a restriction enzyme that cleaves double-stranded DNA at the sequence of GTTAC. Next is insertion. We insert the DNA fragments into plasmid DNA to form a recombinant DNA. Some joined by base pairing, DNA ligase is added to seal them together by forming phosphodiester bond. When two such fragments of DNA cut by the same restriction enzyme come together, they can join by base pairing, while the enzyme DNA ligase is used to unite the backbones of the two DNA fragments, producing a molecule of recombinant DNA containing human and plasmid DNA. Step 4. Transformation and amplification. Transformation of recombinant DNA into bacterial host cell and amplification of recombinant DNA. This is by transferring the plasmid into host cells by transformation process. Treat the bacteria with calcium ions and a brief heat shock to make holes appear in the cell surface membrane. This makes them more permeable to DNA and allowing the plasmids to enter. In other words, insert plasmid into host bacterial cell for replication. Next, screening to obtain the desired clone cell containing the clone gene. 3 methods for screening. Firstly, using antibiotic screening to remove the one with no antibiotic resistance. Using blue-white screening to remove the one with the functional light sac. For non-recombinant plasmid, they contain intact light sac that encodes B-galactosidase, so they can hydrolyze the X-gal and the colonies will be blue. While recombinant plasmids, their light sac is disrupted that cannot be encode B-galactosidase, so they cannot hydrolyze X-gal and the colonies will be colored white. Lastly, nucleic acid probe hybridization. Thousands of recombinant DNA molecules in bacterial cells are screened to find the specific DNA. It is a common approach to detect a specific DNA sequence involves using nucleic acid probe.